Hello everyone, and welcome to the course of NanoBiophotonics, touching our daily life. So, this is the week 6 discussion, so we are halfway down the course, and I am Uditya from IIT Madras. So, coming to this week's contents, what we first understood is so we went through genetic code, then we learned about biosensing background, basics of microarray technology, and DNA microarray and protein microarray. Yeah. So, uh, what we learned about some of the things that we learned here for okay, I'll add one slide. Yeah. So mm, for the genetic code, yeah. So we went through first the central dogma. That this was one of the topics that was handled here. Central dogma basically says how DNA goes to RNA and then to protein, yeah, or more correctly it is mRNA, yeah. So DNA, it is replication. Here it is transcription and translation. So that is the central dogma, yeah. Then we learned about the encoding codes, yeah. What all what are combinations of codons can code for what all amino acids, yeah. So this codon concept, yeah. So it's just like computer codes, yeah. That combination of three letters, how they can command which kind of amino acids it has to code, yeah. So for example, you have a a this means you have to code for lysine, yeah. And then if you have a then it will code for isolysine. So understood how changing the letters, how it will code, yeah. So for example, depending on their application or function, these codes will change, and then that is the concept of gene, yeah. The functional part of your DNA is called as a gene. So that's what then we learned about gene, yeah. How it can have different functional unit of information, and then for example there will be a gene that will be responsible for your eye color, yeah. Such kind of things will be there. So gene is the functional part of your unit, functional unit of your DNA. Then you also learned about mutation. So basically, there are changes in the genetic code, such that genetic code, so that mutation that they can sometimes lead to translation of some other proteins, or sometimes those may not cause mutation, like changes in the protein also. So there are different types of mutations, so synonymous and non-synonymous. So depending on what kind of it is, so if it is synonymous, then the changes won't matter, like the because multiple codons can so multiple codons, like combinations of multiple codons can uh, sometimes give the same amino acid. So sometimes the mutations can be silent, yeah. They won't change the protein or amino acid makeup. Then we learned about different concepts of Human Genome Project and genome sequencing. CRISPR is another CRISPR concept also, which got Nobel recently, yeah, in 2020 in chemistry. So this is a genome editing tool, so genomic Caesar, yeah. So yeah, so uh, yeah. And then if we come to proteins, there will be Protein structures, yeah. Proteins have several levels of structures, yeah. So primary is basically the only sequence of amino acids. Then you have secondary, in which this protein structure are binded either in the form of pleated sheets or beta pleated sheets or alpha helix. Then you can coil it down to tertiary, and then the several coils will come together to form a quaternary structure. So yeah, that's all. Then proteins have different functions, yeah, and folding and misfolding can have impact on it. So this was kind of the overview on the biomolecular things that are possible. Then uh, coming to biosensors. So biosensors. So biosensors usually have three parts. One is the BI, that is the biorecognition unit, placed on a transducer which has a display. So analyte comes and binds to this BRE or biorecognition unit. So yeah, biosensors are important to detect chemical or biological species, yeah, toxins, bacteria, yeah, lot of applications you have seen during the pandemic times, yeah. So but it can go beyond the clinical applications to drug development, environmental monitoring, food monitoring, yeah, a lot of things. So we also learned about different optical biosensors, yeah. Optical biosensors can be of different types, yeah. So they can be based on optical fibers, ring resonators, plasmonic structures, basically gold nanoparticles and such photonic crystals and 2D materials, yeah. So for example, if like if you have gold nanoparticles, yeah, which may be coated with antibodies, upon if these antibodies get bind to some analyte, yeah. So for example, in pregnancy strips, yeah, we use gold nanoparticle based. So gold nanoparticle will be there, and it will be bounded to an antibody. And in your uh, pregnancy strip, there will be another one strip in which antibodies are already there. So this is the analyte. So whenever it will flow, this analyte will form like this, yeah. And because of the red color of gold nanoparticle, you will see a test line positive, yeah. So plasmonic is that's what is an example of a biosensing system. So you have, for example, here we showed antibody, then you can also have a DNA based recognition. So yeah, so there are some, so this immobilization for biosensors, immobilization is one important topic. So physical methods are there, which involves adsorption or spin coating. Then you have chemical methods, which are covalent bonding and such. So each method has their own advantages and disadvantages. So for example, there in the section we also work through optical fiber based sensing, in which this is the core, and then you have clad. So in the surface of this optical fiber, you have something called as evanescent wave, yeah, in this zone, which is kind of which depletes with respect to the distance. Evanescence means that only that it will have a decaying behavior. So if some absorbing species come in this zone, you can see the absorption of the analyte, the surface, yeah. So for example, a gold nanoparticle comes and binds to it, you will see the absorption that gold nanoparticle. Yeah. So several technologies that we saw of microarrays. Yeah. So microarrays is another detection technology. Yeah. So you see DNA and protein microarrays. So DNA microarray is basically a collection of microscopic DNA spots on a solid surface. Yeah. So and you can analyze it simultaneously to check the expression levels. Yeah. So there are several companies like Affymetrix who make this. And several there will be protein microarrays in which there will be a it will immobilize proteins on a solid surface to study protein interactions. So antibody assays or something. So yeah, so in DNA microarray usually DNA is involved, and in protein microarray proteins will be immobilized. Yeah. So some of the important things to check are like surface chemistry, capture agent selection, detection method, yeah, protein extraction. So, if you don't want to work with proteins, which basically have folding effects, you can also go through aptamers, which are basically DNA, short DNA strands, which can work alternatively as a of an antibody to capture protein analytes or small molecules. Yeah. So in place of antibodies, you can go for aptamers, which are more stable and do not have problems such as the folding and misfolding. Yeah. So yeah, they can also go for like they can capture proteins, peptides, cells, and even small molecules, small ions too. Yeah. So going ahead, problems. Go ahead. Yeah. So in the genetic code, one amino acid may be coded by multiple codons. This is due to the presence of so the answer here is redundancy. So this redundancy occurs because the genetic code is composed of 64 possible codons, combinations of 3 nucleotides, while there are only 20 standard. So you have only 20 standard amino acids for which 64 possible codons are possible. 64 combinations are possible. Combinations of 3 nucleotides. Yeah. So there is the redundancy. So uh, yeah, so one amino acid may be coded by multiple codons. Which of the following is an example of a level-free biosensor? So the answer here is SPR. Yeah. So Eliza, hopefully you all know. Yeah, Eliza. What is Eliza? No. So SPRs usually have a kind of a sphere, and you put light, yeah, a laser basically of a defined. So most can be either. So we'll see. So and here it is coated. This sphere, that prism is coated with gold, around 50 nanometer, and you coat antibodies. So depending on this, whatever you will coat on the surface of the thin film, what will happen is this the angle, yeah, this angle, yeah, theta final, yeah, because this you will keep it constant. This angle of reflection will change. So with this, so you can read about the concept. Yeah, there is something called as the evanescent wave generation at the site of total internal reflection. How it excites the surface plasmon resonance on the. So this theta final you can basically monitor using a detector. Yeah, and then upon whenever an analyte will come, yeah, and bind, there will be a further change. Yeah, so you can monitor it using it. So what is label is basically in cases of ELISA. Yeah, so in ELISA what you do is you will have ELISA. So basically it is done in the form of a sandwich. Yeah, you have analyte, and then you will have one uh, antibody in which there will be an enzyme linked immunosorbent ELISA. So because of this and enzyme, you will have a color, yeah, color generated, yeah, in a condition when there will be some subsequent substrate. So basically you are not detecting the analyte directly, you are actually detecting the label. So in such cases it will be a labeled assay. Similarly in polymer chain reaction, it is a there also you will have some molecular beacons which work as labels, and fluorescence based detection is directly when you have like this antibody, you will coat, and then you will have analyte, and then similarly you can have a fluorescence molecule here, so you can check the fluorescence, yeah, and its excitation, and check the emission spectra. So this is here it will work as a label. So SPR is the correct option. The process of synthesizing RNA from a DNA template is called as. So the answer is transcription. Transcription is the process of synthesizing a complementary RNA strand using a DNA template. So it is a key step in gene expression where genetic information is converted from a DNA to RNA, enabling subsequent translation of RNA to proteins. Yeah. What is the purpose of labeling DNA or RNA samples with fluorescent tags in microarray experiments? Yeah. So fluorescence basically helps to increase sensitivity of the microarray. Yeah? Labeling DNA or RNA samples with fluorescence tags in microarrays experiment serves the purpose of increasing sensitivity of the microarray. The fluorescence tags allow for visualization and quantification of hybridization events where the labeled .DNA or RNA probes bind to complementary sequences on the microarray. So this uh, the fluorescence usually give a very good background, yeah, if it is because it is emission of light. So whenever there will be how it has a very good background signal to noise ratio. So it helps in very much increasing the sensitivity of the microarray. Which of the following is an example of an optical transducer used in biosensors? So the answer here is photodiode. So because it converts, as we were asking about optical transducer, yeah. So photodiode converts light signals into electrical signals, enabling detection of quantification of biological events based on changes in light intensity. So the others, yeah, thermocouple basically is involved with temperature. Electrode is involved in the movement of electrons, and piezoelectric crystal is basically changes in vibration, yeah, or mass sensitive basically. Yeah. So any changes in the natural frequency, or maybe on the other way it can also be some changes in the voltage. So yeah, vice versa. Which of the following technology is often used for NGS, next generation sequencing, instead of microarrays? So the answer is RNA sequence, because it offers comprehensive coverage of transcriptome, higher sensitivity, and the ability to discover novel transcripts and alternative splicing events without the need for pre-designed probes. Yeah. So you don't need any complementary RNA probes to get the data done. Yeah, measurement done. Yeah. So yeah, that's a nice method to discover novel transcripts. What information can be obtained from a protein microarray experiment? So the answer will be the abundance of specific proteins in a sample. Yeah. So it can be obtained by labeling proteins with fluorescence tags. As I showed you when explaining the SPR, difference in SPR and how to label with fluorescence tags or other detectable markers. When the labeled sample is incubated on the microarray, the fluorescence signal intensity at each spot corresponds to the abundance of specific protein. So allowing researchers to quantitatively measure and compare the protein levels in the sample. Yeah. So yeah, that's what abundance of specific proteins in a sample. Yeah. Which are the optical properties commonly used in surface plasmon resonance? Yeah, we saw how SPR works. So the answer is reflection. Yeah, we saw about how it is. Yeah, when a specific biomolecules bind to the metal coated surface, it causes a change in the refractive index, leading to a shift in the angle of reflected light. The shift is monitored to quantitatively measure the binding events, allowing level-free and real-time detection of biomolecular interactions. Yeah. So as I told you, yeah, we have a sphere, prism, and you have a laser and detector, which can detect the angles. Yeah. So when you coat it with antibodies, when antibodies come and bind, there will be a change here, in the angle. So theta 1, and then you will have theta 2, and then when you have more, yeah, some antibodies coming, so that they can go to theta 3. Yeah. So for example, change. Yeah. So you can have concentration and theta. Yeah. So this can be the baseline. When something is binding, it will be like this. Yeah. Then you will have maybe washing. Then you can go for baseline, and again it can bind again. So this will be the binding events. Yeah. So this may be the dissociation. Yeah. Whatever is not properly bound, yeah, loosely bound can come down. So you can see all the things. Uh, so not about time access, yeah. In real time basically, yeah, the binding can be in real time. And this height, the magnitude of this binding can give you information about the concentration. Yeah. So that's how you can quantify it. So that's all for today. If you have any questions, you can let me know. Yeah. So I'll be staying. If you have doubts, we can discuss that.